All right, so here's what we're going over today here, guys. I want to talk about a couple of things to do with cell size and their ability to transport material, because we finished talking about diffusion, osmosis, and active transport the other day. So now we need to look at kind of the last thing to do with single cells. Okay, after tomorrow's lab, we'll be dealing with pretty much multicellular stuff. Right? So now we need to look at why can't cells become gigantic? All right, we have to look at what's their limiting factor. Okay, what's the thing that keeps them from being able to do that? All right, the thing that keeps them from being able to do that is their ability to transport material. All right, so what I'm going to need you to do is write down a few of the things that are on the board here. All right, the key points of the three types of transport. Okay, osmosis. It only transports water. All right, so that's an important thing to know. Okay, it's always moving across a membrane. That's an important thing to know. Okay, so it's the transport of only water across a membrane, and it balances solutes or salts. Okay, and it is, of course, passive. It requires no energy for it to happen. It simply occurs on its own. Okay, diffusion is the most commonly used form of cellular transport because A, it's free, and B, works good over small distances. Okay, those are the main two reasons why it is essentially always used by a cell. Okay, the big thing probably being free. Right, and as long as a cell is small, diffusion is good. Right, because we talked about how diffusion works. Diffusion works like how the smell of a fart, okay, gets around an entire room. Okay, if you are very close to the source of the fart, it is very, very strong because the fart gases are highly concentrated at their point of origin. As they spread out, do they become less concentrated? Do the spaces, let's say, between individual fart molecules get bigger the further away from the source of the fart they get? Yes, they do. Okay, and as a result, they don't smell quite so bad. Okay, further away. All right, everybody with me there? So diffusion works like that. If you're close to the source of the material, it's going to seem pretty highly concentrated. But the further away you get, the less concentrated it will be. Okay, so how well would that work in a cell, let's say an imaginary cell, that happened to be the same size as you? Would that be a very effective way to transport material across that kind of a distance? No, it wouldn't. Okay, but across the very, very small distances that cells are involved with, okay, it works just fine. Okay, so we got to remember, essentially, it moves anything within the cell. Once it's in the cell, it gets moved by diffusion. Okay, it's passive, so it's free. Okay, it always moves from high to low, and it is slow. Okay, that's its disadvantage. Now, if it's only going over a small distance, does it matter that it's very slow? No. I mean, if I only have to go one step, even if I take little tiny steps, it doesn't take me very long to get there, does it? Okay, the distance is small. It doesn't really matter that I move slowly. It doesn't take me very long anyway. Okay, the other thing is, it is not targeted. That's what that word is. I think I heard a few people saying, what's that word? It's not any neither this time either. Okay? Targeted means I can't make it go to a specific place. All right? By diffusion, I cannot say, you molecules go over there. All right? It just doesn't work that way. But since diffusion is the transport of stuff from high concentration to low concentration, is it naturally going to go where it's needed? If you need something, it's probably because you don't have very much. Agreed? So it's naturally going to move to the place where there isn't very much of it, from the place where it's being produced, where there's lots of it. All right? So again, over small spaces like the inside of a cell, diffusion works fine, as long as the cell doesn't get too big. Okay? Cell gets too big, then there's trouble. Usually if a cell gets beyond about the 500 micrometers across mark, it's starting to suffer a bit. Okay, you almost never see a cell that's 1,000. Okay, now, could shape affect this? All right, because I'm going to tell you right now, you have some cells in your body that are up to 1 meter long. They're up to 1 meter long. Does that just throw everything out the window I just said? Only if shape didn't make a difference. Okay? Shape does make a difference. Okay? What cells are concerned with, okay, is their surface area to volume ratio. All right? How much surface area do they have? Because their surface area is where they get everything and where they get rid of all their wastes. They want to have a lot of that. Okay? It's important to have a lot of that, and then to have a comparatively small interior volume. Because the bigger their volume is, the longer it's going to take for the stuff they absorb through their cell membrane to get to places within the cell. All right, so have a look at it in terms of this. Here's two different cells. Okay, this is a very big cell. This is a very small cell. Okay, their organelles are the same size. Okay, the nucleus is the same size. Okay, their X's and squares and O's are about the same. They're supposed to be the same size. You get the idea. Okay, they're all the same size. Here's the problem. For the small cell, okay, if it absorbs this blue dot, does it take very long for the blue dot to get to the square? Probably not, right? It's not very far. This cell absorbs the blue dot. Does it take longer to get to the square? Because it's further, right? And they're being transported by the same mechanism, diffusion. So the bigger the cell gets, the longer it takes to move stuff. All right, and that's a problem. All right, think about it this way. Did it used to take a really long time to get through Calgary before Stoney was made? If you tried to get through Calgary in rush hour, there was one way to get through, Deerfoot, the Nightmare Road. Okay, it took forever because everybody was trying to go that way. Okay, and so it got jammed up, and everyone's parked on the Deerfoot, waiting, crawling along at 10 kilometers an hour sometimes. All right, as soon as they built Stoney, what happened? It opened up. Yeah, because now a lot of the traffic can go around. All right, that limits the amount of congestion that can happen on those two main arteries, kind of a thing. Okay, that enabled us to get through the city more quickly. Okay, well, it's kind of like that for a cell. The bigger the city gets, the more ways through it you're going to need. Okay, well, for a cell, it doesn't have that option. There's only one way through the cell. So the cell either has to stay small or it's going to die. Okay, now, do cells naturally grow? Okay, they naturally get bigger. Okay, what do they do when they start to get too big before they die? They split in half. Exactly. Okay, that's how cells reproduce. They start getting too big, like, this is too hard. Split in half. Okay, now I got two small cells again. That's good. Right, now their surface area to volume ratio is a bit better. All right, so does everyone kind of get that idea here? Okay, you can see that as you get bigger, sure, the outer area you have to absorb stuff also gets bigger, but the interior volume gets bigger faster. All right, how many dimensions are there to surface area? If you're going to calculate the surface area of a square, what do? Length times width, okay? What about when you want to calculate volume? Length times width times height. There's 3 dimensions to volume. There's only 2 to surface area. So as something gets bigger, naturally, its volume increases faster than the area around. Okay, everyone follow me there? That's the problem for a cell. It gets so big that the interior volume it's got to feed and transport through becomes too big for the surface area it has to supply it. Okay, everyone follow me on that one? Okay, so I would say quickly write this down because you don't have this anywhere in your notes, okay, and you're going to want it. So, going to move on here, guys, to how this is going to work. Okay, so you've got this sheet in front of you, so you might just want to make a couple notes on here as we go along. All right, the problem we're investigating with this little activity is how does the surface area relate to volume in cells of different sizes? So we want to see, does your surface area to volume ratio change, okay, as you get bigger? Now, we've kind of already said that it does, but we want to kind of mathematically prove that. All right, we also want to look at, okay, here's a single-celled organism. Here's a single-celled organism. So micro is single-celled. Macro is single-celled, but macro is a lot bigger. Okay, we want to see, does the surface area to volume ratio get really volume heavy if you're a big single-celled organism? Then we also want to look at multimicro. Okay, multimicro is supposed to represent a multicellular organism. So we're multicellular, we're big, but our cells are all still very small. We want to see, does that give you an advantage? Does that allow you to get big without having to deal with the problem that, let's say, macro is probably going to encounter? Okay, is your surface area to volume ratio going to stay good if you're multicellular versus single-celled and really big? Okay, Cam, question? No. Once you get to macro, the surface area to volume ratio is going to get blown out of the water. Okay, it's just not going to work. All right, so the hypothesis we need to generate here, okay, generate a hypothesis about the relationship between surface area and volume in cells and how this can affect the functioning of the cell. So in the if part, okay, tell me as the cell gets bigger, okay, the and part, we're simply comparing some hypothetical organisms here. Okay, the then part, tell me what's going to happen to the functioning of and the transport within the cell. Okay, so that's what we're looking for our hypothesis. That's pretty much a dead giveaway there. Everybody should get 4 or 5 out of 5 on their hypothesis now. All right. So what we're going to do is we're going to make a table, and I'm going to show you what that table will look like in a minute here, and we're going to calculate the surface areas and volumes of these cubes. All right, and then we're going to make a surface area to volume ratio. Okay, everybody with me so far? It's easy math. In fact, we're going to do it together. All right, so everyone's going to get the math right. All right, so here's our calculations. Okay, we've got our cubes down here, and this is the chart. You're going to need to make this chart in Google Docs, okay, at some point. But you're going to have the cube, micro, macro, and multimicro. The surface area, which we'll calculate here in a minute. Okay, the volume, and the surface area to volume ratio. Now, if I want to calculate the surface area of a cube, how do I do it? Nope, that'll give me the volume of the cube. Length times width times height will give me the volume. A side cubed, no, because that's still going to give me cubes. Okay? If I just go 1 cubed, I won't necessarily get the surface area. I'll get the volume again, right? No. How many sides in a cube? 6. Okay, so there's 6 sides on a cube. Can I calculate the surface area of one side of the cube? By going length times width. And then I multiply that by the number of sides. Agreed? Length times width times the number of sides. That'll give me the surface area. All right, because I'm finding the surface area of one side, one square, and there are 6 squares on the cube, so I multiply it by 6. All right, so to get surface area, we're going length times width times 6. All right, these are not hard calculations. All right, for micro, it's 1 times 1, which is 1, times 6. What's 1 times 6? 6. Okay, so that's easy, right? Okay, we're going to say it's 6 micrometers squared. All right, if I want to calculate the volume, it's length times width times height. What's the volume? 1, yeah. Except now it's in cubic micrometers. So the surface area to volume ratio of little single-celled micro here, okay, is 6 to 1. Is that good? Actually, that's really good. Anytime their surface area is bigger than your volume, ratio-wise, you're good. Okay, especially 6 to 1. That's like kind of ideal. It's a U with tails. it's go insert symbol, and you'll find it in the symbols. It's the Greek letter M. Right? Okay. For macro, and if you can't find it, you can just write a U. I'll know what you mean. Okay. All right. For macro, same thing, except now our sides are 10 times as big. Okay. So if I want to find the surface area of macro, 10 times 10 times 6. Okay. 10 times 10 is 100. Times 6 is 600. All right. UM's squared. All right. I want to find the volume of this one. Okay, 10 times 10 times 10 is 1,000. All right, this one was 6 to 1. What's this one? No. Okay, 60 to 100, yes. Or if I divide both of these numbers by 1,000, do I get this? Is that significantly worse than the surface area to volume ratio for micro? Yes, it is. Okay? Same shape, but I made it 10 times bigger. And by making it 10 times bigger, I made it way worse off. Okay? Now the surface area to volume ratio is 0.6 to 1. Okay? That is much, much worse. We do not want that kind of a number. All right. Now we got to look at multi-micro. It's kind of the tricky one to calculate. All right. Now, it's exactly the same size as macro is. So does it have the same volume? Yes, it does. It has the same volume as macro because they're the same size. They take up the same amount of space. Okay. So the volume is still 1,000 micrometers cubed. But does it have the same surface area? It does not. Okay. Because rather than being hollow, it is made up of many little micros. Yeah, it is. It's going to have a way bigger surface area because each and every micro has how many sides? 6. Okay? So even though they're not on the outside, we still count that surface area because not all of your cells are on the outside, are they? Okay? They're everywhere, but we still count their entire surface area. All right? So each one of these micros is exactly the same size as this one. So if I want to find the surface area, can I take this surface area, 6 micrometers, and multiply it by the number of micros that are in here? That's right, 6,000. There's 1,000 micros in this thing. Okay, so the surface area here is 6,000 micrometers square. So what's the ratio of surface area to volume for multi-micro? 6 to 1. Okay? So as a multicellular organism, you have the same surface area to volume ratio as a single-celled organism, and that makes you efficient. Okay? But it is the stumbling block to having a giant amoeba sitting in that desk. Okay? A giant amoeba would only have one cell, and it would be unable to transport material effectively. Oh, it'd be a lot harder. You'd have to use a micrometer and stuff. You could do it, but that's why we used cubes, to make our life easy. Okay? All right, so you got that chart there. Okay, it's going to be on the podcast. If you didn't get it, you can catch it later. Okay, so here's what we need to do with those numbers. Okay, you got these questions here that you're going to have to answer. Okay, there's 7 of them. The 2 are on the back. Okay, on yours they're on the back. So don't forget 6 and 7 on the back of your sheet. All right, so for 1, 2, and 3, it's basically tell me about some stuff that's from your chart. Okay, so for number 1, it says when the cube dimensions go from 1 to 10, that's from micro to macro, the increase is a factor of 10. But how many times greater does the surface area get? Okay, so what you got to do is go, all right, well, the surface area of macro is 600. The surface area of micro is 6. How many times greater is that? Put that in. Okay, that's what that one's asking. I'm not going to give you that answer, but I think you can figure it out. Okay, and then it asks same thing for the volume. All right, well, the volume of micro was 1. The volume of macro was 1,000. How many times bigger is that? Okay, that's what that question is asking. Okay, then for number 2, for the 2 cubes that are big, macro and multi-micro, they have the same size. That's what this question is saying. It says the size and the volume have a factor of 1. It means they're the same. Calculate the factor for the difference in surface area between them. All right, so we go back to here and we go, okay, the surface area of multi-micro is 6,000. How many times bigger is that than 600? Okay, whoa, that's for you to put in, not for you to shout out. Okay, all right. Then for number 3, for which two cubes does the ratio between volume and surface area change when they get bigger? All right, so we go back to this and we look at it and we say, okay, if micro gets bigger, it essentially turns into macro. Would that change its surface area to volume ratio? Okay, if I made macro bigger, it would be an even bigger cube with an even more poor surface area to volume ratio. But if I made multi-micro bigger, what does it fill up with? More micros. Does its surface area to volume ratio stay the same? That's what that question's asking. All right. And then for number 4 is asking about these boxes here. Okay. Now, you're going to have to measure these on your sheet. You're going to need a ruler for this, and you're going to have to measure the sides of these shapes. What this question is asking is, these all have the same volume, or at least very close. Okay? But is their surface area to volume ratio different because they have different shapes? So what you have to do is find the surface area of each one of these, okay, and see if it's significantly different than the others. Okay? You don't really need to find the volumes for these because we're saying that the volumes of them are the same. Okay? So find the surface areas and see, does one of these have way more surface area than the others? If it does, then shape is a factor. Okay? This is like this cell here. Okay? This is a nerve cell. These are the ones that in your body can be up to 1 meter long, yet they still function. Okay? Shape makes a difference. You got to prove it to me mathematically in question number 4. All right? Everybody with me there? Now, when you're calculating the size of these, okay, or sorry, the surface areas, remember that not all the sides are the same. All right? So for calculating the surface area for B, you got to calculate the surface area of this side and multiply it by 2 because the back is also the same. Okay? Calculate the surface area of the top, multiply it by 2 because the bottom would be the same. Find the surface area of this side, multiply it by 2 because the other side would be that, then add them all together. That'll be your total surface area. Okay? Same thing for C. For A, it's essentially a cube. Okay? So you can just calculate the surface area here and multiply by 6. Okay? Everybody with me for that one? For number 4, so there's a little bit of math, but it's not too difficult. Now number 5, like we said, number 5 goes along with number 4. Here's 3 different cells in your body. A liver cell is like A. Skin cells are like B. Nerve cells are like C. Okay? Tell me, how have they adapted to where they are in the body? Can they function where they are? Okay? Based on how they might be supplied. Okay? In number 6, okay, we got a picture of a red blood cell here. Okay? So here's what a red blood cell looks like. Okay? Here's what a red blood cell looks like super enlarged. They have this concave, biconcave shape, right? They look like a donut head on, but from the side, they look like this. Okay? Does that give them more surface area than if they were shaped like that? Okay? That's what number 6 is asking. Does the shape help them to do their job? What's the job of a red blood cell? Transport oxygen in your body. Can probably transport more with a bigger surface area. Something to think about. Okay? Number 7. Most materials get from place to place in the cell by diffusion. Okay? What effect would a low surface area to volume ratio have on intracellular transport? So if you got a bad surface area to volume ratio, what's it going to do? That's what question 7 is asking. Okay? Then you got your conclusion, and you're good. You do not have to do any variables in this lab, because there aren't any. It's not a lab. Okay? It's all theoretical. There aren't any variables here. Okay? So you don't have to do a design. Okay? There's no evaluation either, because there's no sources of error. Okay? So it's a little bit shorter. Okay? You got hypothesis. Okay? You got your procedure, which is basically the chart we filled out together. So that's easy 5 marks. Okay? And then your analysis, all right, which is basically your questions here, your answers to them, conclusion, and then overall. Okay? You've still got 20 minutes. Okay? To work on this, I'll put this chart back up so that everyone can have it. Okay, get as much of it done as you can right now, and that will limit your homework. This is going to be due on April the 3rd. Okay. Yeah, you don't have to write out the problem again because there's no design, and that's where I mark the problem is in the design part, so you don't even have to worry about the problem on this one. Okay? So it's a lot shorter than a normal lab report. So you got your microscopy lab that we finished today, due April 1st. And you've got this thing that we're doing today, due April 3rd. Tomorrow we're doing another lab. You have a lot of stuff coming up in the next little while. Okay? Make sure you stay on top of it all. All right, get to it.